The broadcast is now starting. All attendees are in listen-only mode.
Good morning or afternoon for some of you, and welcome to Xenotech's webinar, Application of Hepatocytes for In Vitro Uptake Assays. My name is Matt Beck, and I am the Marketing Manager here at Xenotech. Today I am pleased to be joined by Xenotech's Chief Scientific Officer, Dr. David Buckley. Before we get started with today's presentation, I would like to go over a few housekeeping items. Here is an example of your GoToWebinar user console. Using the orange arrow will allow you to minimize and maximize the user console. Please note that your audio is muted. If you have questions during the presentation, please post your questions in the questions pane seen highlighted here. We will have a Q&A session immediately following today's presentation to address questions posted via this questions pane. A copy of the slides and recorded session will be available a few days after the presentation. We will send both via links through email for your reference. So with that, I am pleased to have Dr. David Buckley here with us today. David received his Ph.D. in toxicology and pharmacology from the University of Kansas Medical Center. After completing his doctoral training, David joined Xenotech as a member of the research and development team and currently serves as Xenotech's Chief Scientific Officer. David's research focuses mainly on hepatic drug metabolism with an emphasis on cytochrome P450 and human-based in vitro systems for the preclinical evaluation of metabolism-based drug interactions. David has published several peer-reviewed journal articles in the fields of drug metabolism and toxicology and is a member of a number of scientific societies, including ISSX. It is my pleasure to present to you Dr. David Buckley. Dave?
All right, thank you, Matt. While I set up here on the screen, I just want to reiterate a couple housekeeping items that Matt pointed out. Hold on one second. I'd like to reiterate that the slides will be available following the seminar. We'll probably have them up on our website available to you within 24 to 48 hours from the time we deliver this. So if I'm moving through information too fast, don't feel like you have to take notes, because this information, again, will be available. At the very end of the presentation, we'll have time, hopefully, for a few questions. You can put those, again, in your dialog box, and we can hopefully get to those at the end. If we cannot get to your questions or you have other comments or suggestions or information you'd like to receive from us, again, my direct contact information will be available here on the slides, and, of course, you can also contact Xenotech directly. Today we're going to talk about the application of hepatocytes for in vitro uptake studies. My main goal today is to provide a very high-level overview of all the different applications of both suspended and plated hepatocytes for use in these uptake studies. And it's a quite extensive overview of quite a bit of literature, some of which I had to pare down and make some assumptions, but we'll work through that today, and I'll try to point out all the relevant information where possible. So just an overview of today's topics. I'll review some of the basic concepts, particularly a structure-based approach to looking at uptake. I'll talk about the selection of appropriate in vitro study type and design, so what test system and what type of study design would be adequate. Description of the basic in vitro methodologies using suspended hepatocytes, primarily looking at the oil spin method, as well as the whole system versus medium loss method. And also talk about looking at uptake into plated hepatocytes. And with each of these, I'll talk about some of the practical challenges that you may have to overcome when doing these experiments in the lab. I'll talk a basic review about the estimation of active and passive uptake and the ability to predict active uptake clearance. I'll talk briefly about the estimation of fraction unbound hepatocytes, at least the point of view from a couple of recent manuscripts or papers that have been recently published, as well as I'll talk about some of the prediction techniques for comparing your in vitro data to predict in vivo clearance. Again, today's purpose is mainly a high-level overview, so I apologize if I don't get into all the details. I know there is a plethora of information that is available, so unfortunately we won't have time to cover it all today. Most of you are probably aware that the recent regulatory changes, both in Europe and the United States in 2012 in regard to DDI. This new guidance from the EMA and FDA came out in June and February respectively. And one of the major changes had to do with the incorporation of transporters, and that has been highlighted for looking at transporters that are present particularly in the gut, in the GI tract, the liver, kidney, blood-brain barrier, amongst others. And this has really put an emphasis on understanding hepatic uptake and what active transporter processes may be involved for uptake of drugs into hepatocytes. The recommended hepatic transporters, today's presentation is going to be focusing on hepatic uptake. So the three that I'll primarily mention are your organic anion transporting polypeptides 1B1 and 1B3, as well as OCT1. These transporters pump drugs from the blood, from the sinusoidal membrane, into the hepatocyte. What I won't cover today in too much detail are some of the efflux transporters localized to the cannulicular membrane, such as BSEP, BCRP, and PGP, and MRP2. So the main perpetrators that we'll talk about today in regard to the different transporters, again, are the OATPs and OCTs. And a lot of the experimental designs I talk about will be evaluating compounds that undergo active transport by these three transporters. We start thinking about structure and physicochemical properties and how that's relevant to hepatic uptake and even metabolism. So we're evaluating new drug candidates when they first come into Xenotech, and we're asked to predict whether or not they may undergo transport and/or metabolism. Some of the physicochemical properties that we typically ask for are the log P, or log D 7.4, the polar surface area, and the pKa. The log P or log D is a measure of the lipophilicity, and this will, so drugs that have a high log P will have a high passive rate of diffusion, or typically can cross membranes fairly freely, but also require a biotransformation step in order to be eliminated. We'll look at polar surface area. If the polar surface area is less than about 50 angstroms square, then the drug candidate will likely be completely absorbed. It'll be able to permeate membranes fairly freely. Whereas if it has a polar surface area of at least over 150, but even oftentimes you start approaching over 100, then you're going to need transport to basically be able to cross the membrane. So these drug candidates potentially, if they're getting into certain tissues, may be a candidate for as a substrate of a drug transporter. And of course pKa, so we're looking about ionization of particularly amine and acids. So depending on the pH for which the compound is in, whether it be gastrointestinal or more of a blood-based physiological pH, these acids or amines can potentially be ionized or unionized, and of course the unionized form is available to freely transverse membranes, whereas the ionized form will typically require transport to be able to cross those membranes. So again, these are some just generic principles that we typically look at regarding physicochemical properties and whether uptake and/or metabolism may play a role in the clearance of these drugs. If we take a look again at the phys-chem properties and evaluation of hepatic uptake, there are potentially several causes of a high liver to blood ratio. So instances where we have uptake into the hepatocyte in which the concentrations in the hepatocyte are going to be greater than what we see in the blood. For acidic drugs, when this occurs, we're typically thinking about active uptake. And the transporters that we're considering there, particularly for anionic drugs, are OATP 1B1 and 1B3. Examples of these types of drugs are statins as a class. In these cases, we'll want to evaluate the drugs as substrates for hepatic uptake transporters. If we have a basic drug, a cationic drug, these can go into two different classes. We can have lipophilic amines that are relatively unionized at physiological pH, and these undergo rapid passive diffusion and can also undergo lysosomal trapping. So this is essentially pH trapping in the lysosomes of the subcellular compartment. Many of the cardiovascular and CNS drugs, particularly antidepressants, undergo this lysosomal trapping. But the way that the compounds move into the cell is a passive diffusion. We have compounds that undergo active transport into the hepatocyte, particularly for hepatic OCT1. These are going to be typically compounds that are organic cations, so they may be charged at a physiological pH. Again, we can evaluate these drugs for substrates for lysosomal trapping or active uptake using hepatocytes or other types of cell-based systems. Just a generic overview of some of the methods that we can use to evaluate uptake into hepatocytes. There are three common in vitro methods. By no means is it limited to only these three, but if you look in literature, these are probably the three that you'll find most common. The first is use of an oil spin method. And this slide is just going to be an overview, and I'll describe these methods in detail on some subsequent slides. The oil spin method, we'll use suspended hepatocytes. We do a rapid uptake incubation, and we'll isolate the hepatocytes from the medium by centrifugation through a silicone oil layer. If we look at the medium loss method, we're going to look at loss from the medium versus the whole cell system And again, this is typically done in suspended hepatocytes. However, this methodology can also be applied to plated hepatocytes to look at medium loss. Here, if we're using these in suspension, we'll centrifuge the hepatocytes after the incubation, look at loss of drug from the medium, or by looking at loss of drug from the whole system. And we can also, again, use plated hepatocytes. So these are going to be attaching whether human or non-human hepatocytes. Very similar techniques to what we do for the oil spin, as far as we'll look at the uptake of a compound, relatively short time points under initial rate conditions of the drug into plated hepatocytes. We can wash the cells after a particular incubation period. We can evaluate uptake into the cells, or we can also measure the loss of the drug from the medium. All of the methods I mentioned above can be employed to evaluate active uptake by transporters, and lysosomal trapping, as I mentioned, which is more of a passive process, but it's a partitioning event based on pH. These can be evaluated by the medium loss or plated cell uptake methods. If you want to see Xenotech's website, we have some posters in a recent publication in D&D indicating these methods for lysosomal trapping. But again, today's focus, I'll primarily be talking about methods to evaluate active uptake by transporters into hepatocytes. So there are various hepatocyte formats that are going to be available. So if you go to a vendor, you're typically going to see various types of suspended hepatocytes and plated hepatocyte products. This slide is really going to be to break down what are the common applications of each of these different products, and what do you typically see when you get a product characterization sheet, whether that's from Xenotech or even from another hepatocyte vendor. So there's various applications. Of course, product characterization, we can determine uptake, whether that be an active uptake or whether it be passive. We can look at transporter phenotyping. We can also evaluate inhibition potential for drug candidates. We can evaluate intrinsic clearance and potentially scale that to in vivo. And we can hopefully also determine what is the rate limiting step between metabolism and or transport. And just some common utility of these different hepatocyte products. Hepatocyte suspensions, which can be either an individual or pooled, and there's various pooled products that are available on the market. We can do these with the oil spin method and carry out most, if not all, of these different applications. The two products that Xenotech currently has on the market are our Hepatosure, our Pool 100, as well as our CryoStax, which is our single freeze pool. We also have hepatocyte suspensions that can be used for the medium loss method. Again, it's going to be the same products, and again, these are pooled products I have listed here. But medium loss methods have been primarily used to determine intrinsic clearance, as well as the role of the difference between metabolism and transport. And lastly, plated hepatocytes. These are going to be using attaching hepatocytes, whether they be from human or non-human species. Again, plated hepatocytes can be used for all these various uptake methods, as well as potentially looking at efflux transporters as well, which won't be discussed in detail today. One thing I will point out is that the selection of the test system and the methodologies will be highly dependent on the application and the purpose of the experiment. So what are the true outcomes and parameters you're trying to define? What will help us be able to select the appropriate test system and appropriate experimental conditions and methodology to get to the endpoints that you desire. So I'll talk basically right now about the basic methods for uptake into hepatocytes. This is simply asking the question, do we have uptake into hepatocytes, yes or no? And again, we can use all three methodologies. We can use the oil spin method. And the oil spin method, again, is going to be using suspended hepatocytes, incubated typically with a single concentration of substrate, typically to low concentration. And we'll do this for one or more short time points. And typically we want to capture things under initial rate conditions, which is going to be within your first minute, maybe out as long as even 2 minutes. So we're going to try to capture these very short time points. We can measure this uptake by evaluating either hot or cold compounds. We can do this by scintillation counting or by LCMS-based approaches. Medium loss, so we'll do an incubation again primarily in suspended hepatocytes. After the incubation time points, we'll centrifuge to determine loss of drug from the medium itself. Like the oil spin method, we'll use a single low concentration of substrate. We'll look at disappearance from the medium. Also, we can evaluate metabolic stability of the compound by monitoring whole system loss. And for plated hepatocytes, again, experimental design is going to be similar to that of the oil spin. A single concentration, look at one or more short time points. Hepatocytes can then be washed after these time points, and we can evaluate the uptake of drug into the cells and potentially even the loss of medium. So these are the basic uptake methods that are built, ability to give us an approximation of a rate, initial uptake rate for certain substrates. And when you're buying products from vendors, whether it be Xenotech or another vendor, these are the typical types of assays, the basic type assays that are done to characterize products. With each of these methods, we can also evaluate the active versus passive component. Some potential ways to do this are using a hot test article, radio-labeled compound. We can evaluate the hot compound in the presence or absence of a molar excess of a cold test article. And if we're evaluating this with cold compound, where we're doing the analytical analysis by LC-MS, we can look at active and passive component at 37 versus 4 degrees, or we can look in the presence or absence of specific transporter inhibitors. Now I'll go through each of the methods I've described in detail and to show you some of the basic flows of each of these experiments and some of the examples of data that may come out. So if we first take a look at the oil spin method, it's pretty simple. So we can use a plastic tube. We'll fill this with a silicone oil with a specific density, and below that we can have sodium hydroxide. We can do an incubation with our hepatocytes, and at each time point we can put the substrate and hepatocyte incubation mixture on top of the silicone oil layer. Again, this is listing radio labeled, but this can also be conducted again with cold compound. We can then centrifuge the hepatocytes to the silicone oil layer. It'll separate, only allow the hepatocytes to go through, and the free drug in the medium will stay on top. Once the centrifugation has occurred, we have pellets that have now been formed. We can now freeze the tube, cut the tube in half, be able to collect the bottom half with the hepatocytes and the drug taken up into those, and measure that by whatever analytical methods we deem necessary. We can also look at what is left in the medium by analyzing the top of the tube. So just an example of some data generated with hepatic uptake with the oil spin method. Here we use our pooled human hepatocyte product, our pool of 100. And we just did a simple experiment to determine active versus passive uptake. The experimental design, we used a single low concentration of substrate. In this case, it was 1 micromolar ester 3-sulfate as an OATP 1v1 substrate. And we did this at several time points. We focused many of our time points around those early time points up to 90 to 120 seconds within the first 1 or 2 minutes to try to capture the initial rate. We also did in this experiment, we did this out to 20 minutes, so you're able to see some of the data here. We did this at 37 versus 4 to be able to differentiate the active versus passive components. We were able to determine the initial rate conditions, linearity with time. So one of the things I will point out, which is a practical limitation, is in many of these assays, the linearity is often lost in as little as 60 seconds or less. And even these data we show here that after about the first 2 time points of 50 and 30 seconds, that even after 30 seconds we start to lose some of our linearity with time. So as a practical challenge, that's something to take note of, because when we start talking about determining clearance later, it's going to be important to look at uptake rates and our initial rate conditions. With this, we can calculate our uptake rate. Again, we'll try to do this under like a 1-minute time point, as short as possible. And we'll look at, typically be expressed, at least for suspension, in peak moles per minute per million cells. Next, I'll move on to the whole system loss versus medium drug loss. Here we'll do an incubation of a drug, again typically a low single concentration of substrate, anywhere maybe from about 0.1 to 1 micromolar. And we'll do this again in suspended hepatocytes, again typically maybe 1/2 to 1 million cells per mil, potentially lower, potentially even higher. We'll let this incubate again for short periods of time, in this case anywhere from 15 to 60 seconds up to 1. And we can then look allow the compound to either equilibrate into the cells or be actively transported into cells. And after each time point, we can put it through a centrifugation step. We can spin down the hepatocytes. We can now sample out the medium to look at medium loss. If we look at just the whole system loss, so what we'll do here experimentally is at our time point, we can actually pipette out a sample of the cells that are cell in suspension, drop that into an organic solvent such as acetonitrile. And what we're going to stop any uptake reactions. We're going to basically lyse the cells and stop any metabolic reactions. And what you measure here is you're going to be measuring drug loss that can only occur by metabolism. So the only way you're going to lose drug, parent drug, is by metabolism in this process. So this is looking at, this is kind of your standard metabolic intrinsic clearance type assay. If we look at medium loss, so loss of the drug from the medium, drug loss can occur by two mechanisms, so both uptake into cells, whether it be passive or active, as well as metabolism. So combining these two together, we can get somewhat of an idea of the relative rates of metabolism as well as metabolism plus uptake. So looking at drug depletion profiles with the medium loss method, this helps us again identify the potential rate limiting step in uptake or metabolism. An example of this from a recent publication from Brian Houston's lab is looking at fexofenadine and rat hepatocytes. In this case, the metabolic stability or metabolic clearance is highlighted here by the black circles. So this is looking at whole system loss. And the open circles represent uptake. So this is going to be your medium clearance, so your loss just from the media, which again is representative of both uptake and metabolism. In the case with fexofenadine, the metabolic clearance is going to be less than the active uptake clearance, and basically showing the relationship between those two. If we take the drug clarithromycin, again in rat hepatocytes, again this is in the same publication, in these cases these two lines basically mirror each other. So in this case the metabolic clearance is equal to or exceeds that of the uptake clearance, and whether that's active or passive was not, at least not determined with very basically with this set of data. The metabolic clearance here is relatively fast, at least in comparison to metabolic to the uptake clearance. In this case, the medium clearance, so whether it again be about passive or active mechanisms, appears to be the rate limiting step. Next, I'll move from suspended methods over to plated hepatocyte methods, and we can look at active uptake with plated human hepatocytes. The same rules and general procedures will apply to non-human species as well, such as rat or dog or monkey hepatocytes. Our typical test system, again, we plated hepatocytes, and we can allow them to adapt to the substratum for anywhere from, typically what you'll see in protocols is 6 to up to 48 or more hours. Sometimes you can see as little as 2 or 4 hours to allow for attachment, all the way out to maybe 4 or 5 days in culture. The main thing you want to allow for is allow enough time for the hepatocytes to actually adhere to the substratum. That way when you do a wash step, that you're not actually sloughing off many of the hepatocytes, because that could greatly skew your data. The benefit of potentially doing these for the very short term is, one, you can do them potentially in the same day, as well as the ability for the hepatocytes to resynthesize and relocalize efflux transporters may be somewhat negated at these very short time points, where that has to be taken into consideration for multiple days in culture. Some protocols also call for an incubation in serum-free media, so maybe allow attachment for 4 to 8 hours, incubate for a few hours with serum-free media, and then to replace media with serum-free media that contains your drug candidate or test article of choice. Again, there's various different iterations to this, but again, these are just different parts to consider when you're setting up these different types of assays. So just a basic overview of an assay type. Typically, if we're doing just a basic assay, again, we'll run a single low concentration of substrate, several time points, particularly within the initial rate, within the first minute or two. We can do this in the presence or absence of transporter inhibitors. We can also do this in the presence or absence of cold compound if we're using a rate-labeled test article. We can remove media at each of the time points, wash cells. We can then lyse cells and measure uptake of drug into those. Determine initial rate velocity. We can calculate the uptake rate, and again, this can be expressed in multiple ways. So picomoles per minute per number of cells, or you can even do a BCA and classify this on per milligram of protein basis. I'll move from some of the basic approaches that we're looking at, just a simple yes or no answer for uptake to those methods used to estimate uptake clearance. And I'll talk about some of the basic parameters and assumptions that will be relevant for many of the next few slides. So our typical test system has hepatocytes. It's going to be represented by this blue circle here on the left. These again can be in suspension, and as well they can be in hepatocyte, plated hepatocytes. And the red circles over here represent just free drug in the medium. The common uptake mechanisms that are typically evaluated in many of these models are going to be active uptake, which is registered here, which is typically going to be due to an active transport mechanism such as OATPs or OCT. You can also have passive diffusion, which again is going to be highly dependent on your physical chemical properties and the concentration gradient, but this is also a force that can drive the compound into the cells. So we have cellular properties and metabolism. So once the compound gets into the cell, we can have partitioning or sequestration within certain subcellular components, non-specific binding to components and proteins and lipids within the cell, and that must be taken into consideration. As we can also have clearance from the system by metabolism, so metabolic clearance. Many of the protocols and publications that you'll see will do this either in the presence or absence of 1-ADT, which is a pan P450 inhibitor. This will be used to knock out metabolism to allow compounds to equilibrate without having extensive metabolism. At least in one or two publications that I've observed, and I'm sorry I can't remember this off the top of my head, 1-ABT was evaluated for the potential to inhibit some of these transporters. I think had little to no effect, at least at the concentrations examined. And also we have potential efflux mechanisms. If you're using suspended hepatocytes, your active efflux is assumed to be somewhat negligible due to internalization of efflux transporters. And there are a handful of publications that demonstrate this. If you're using plated hepatocytes, particularly plated hepatocytes that have been a number of hours or days in culture, then active efflux may be a component that cannot be assumed to be negligible. And we also have, just so we have passive diffusion in, we can have passive diffusion right back out of the cell as well. And again, these will be parameters that will be common for most of the methods I'll discuss over the next few slides. So the methods for evaluating clearance from hepatocytes, we can use all the methods I described before: oil spin, medium loss, and plated hepatocytes. With the oil spin method, how we're going to change this up is now we're going to be using suspended hepatocytes in the presence of multiple concentrations of substrate. And this will be done at multiple time points, particularly those to capture, again, the initial rates, so the first minute or two of incubation, as well as oftentimes we'll look at time points clear out to as long as 45 minutes to 90 minutes to allow for equilibration to really get a good feel of the medium to cell ratio at equilibrium or steady state. We can get the total uptake intrinsic clearance. We can define the active uptake clearance components, the passive diffusion component, and evaluate the fraction unbound in the cell. And with some of these methods, if you take them into consideration with other measurements, such as the measurement of the medium of the cell, you can eventually arrive at an estimation of the free intracellular concentration in these experiments as well. If we look at the medium loss method, again, here we're going to centrifuge hepatocytes to determine loss of drug from the medium. Typically, this is still done with a single low concentration of substrate. There is some potential utility to doing multiple concentrations of substrate using the medium loss. But a typical method typically incorporates a single. Again, we can look at the medium loss to determine clearance of disappearance from the parent. We can also evaluate the metabolic clearance by looking at whole system loss. And again, this is primarily to evaluate metabolism and transport. Plated hepatocytes, again, be very similar experimental design we saw with the oil spin method. The biggest difference between the basic methods and these clearance methods are incorporating multiple substrate concentrations over multiple time points. And again, will determine the active, passive components, fraction unbound, as well as a variety of others. And again, most often these experiments consider using a pan P450 inhibitor such as 1-ABT. Again, and depending on the application, the model you're building, whether or not you want to incorporate metabolism or clearance of some of the metabolites, this may or may not be useful for your experimental design. One of the first applications of the medium loss that was published by Soares et al. In 2007, and this was predicting using the medium loss method to better predict in vivo hepatic clearance and whole system loss, mainly in regard to their previous experience looking at whole system loss, which is a measure of just metabolic clearance. So using the medium loss as a measure of both uptake and metabolism to better scale your in vitro to in vivo results. This study, it's been highly cited. I've seen it in numerous presentations around the world. They looked at 36 test compounds for metabolic stability in rat hepatocytes, examined the loss in cell culture medium versus whole system. Here's some example data. But the real meat of it was here in the prediction of the in vivo results from the in vitro data. If they use a conventional metabolic stability method looking at this whole system loss, the in vitro, as we know, will underpredict the in vivo, and that's shown here in this experiment. We go over to the graphs in the middle and the graph on the right. Now we're incorporating the uptake clearance as well as just the metabolic clearance. So we're looking at medium loss. And this gave a much better prediction, at least in these rat hepatocytes with these model drugs, much better prediction of the in vivo clearance from your in vitro data. So this was a major improvement in this area. If we look at active uptake with the oil spin or plated methodology, if we're looking at clearance, we can do this with a conventional two-step approach, and there's as a number one, and number two that I'll talk about as a common method more recently is using a simultaneous fit with a two-compartment model. I'll talk about that here in some subsequent slides. But the conventional two-step approach, determining the active and passive uptake, so we'll evaluate multiple concentrations of your substrate, multiple time points, and these are typically, again, done very early time points. And we'll do this at 37 degrees and 4 degrees Celsius. We can get total uptake at 37, our passive uptake at 4, and we can derive our active uptake component from that. So we'll determine the uptake rates under initial rate conditions. We can evaluate what our passive uptake rate is as well. We can determine the uptake kinetics from the initial uptake velocity, so derive our Km and Vmax values as well as our passive diffusion component. And this can be done with relatively straightforward and simple programs such as Graphit. From this, we can determine the active uptake intrinsic clearance from basically just dividing your maximum velocity, your Vmax, over your Km values. And again, from this value in your PDIF, you can do several things which I can talk about later. But this is, again, just a summary of this broad approach. A little bit more mechanistic is looking at the two-compartment model, which requires a simultaneous fit. So here you're determining active versus passive uptake. Again, multiple substrate concentrations at several early time points. You have the potential to only run this at 37 degrees with 4 degrees being an option. And again, this can be done in the presence or absence of a metabolism inhibitor. From this, we can get our total uptake. We can eventually derive our, subtract out and derive our passive component, our PDIF, and subtract out and derive our active uptake component. In addition to looking at the short time points, many of these protocols and experimental methods call for looking at the cell to medium concentrations at steady state. So basically looking at the ratio of the cell to medium at equilibrium. Therefore, that's why these 30 to 90 minute time points, these longer time points, are typically included in these types of methods. You can process these data. You can fit the model with a simultaneous fit with a model. Put these models in a program like MATLAB. We can establish multiple parameters for uptake. We can determine our Km Vmax, our passive component. We can derive our fraction bound in the cell, the concentration of drug in the cell, the unbound concentration of drug in the media, and those can be measured as well. And we can also look at the metabolic clearance if necessary. So there's many parameters you can derive, and I'll talk about using some of these parameters in some subsequent slides. Now if we think about the active versus passive uptake into hepatocytes and what are the impact of the active versus passive clearance mechanisms as we increase concentration of the drug. An example of one would be for pitavastatin, where our active uptake clearance is about 10 times what we see of our passive diffusion rate. So here at low concentrations of pitavastatin, active uptake represents approaching 90% of uptake into the hepatocytes. But as we increase pitavastatin concentration, the passive component starts to increase and active component decreases. You start to saturate and you also start to drive up the passive component. Kind of conversely to this is sequinavir. So here the active uptake clearance and the passive diffusion rate are approximately the same. So it's about a 50-50 mix. And as you drive up your concentration, eventually your passive component will start to diverge from your active component. So as you're doing these experiments in vitro and evaluating the active versus passive component, one important thing to keep in mind is when you're thinking about what may be a pharmacologically or physiologically relevant concentration of your drug candidate at the hepatocyte, which of these mechanisms are going to predominate based on the concentration that's available. So again, with pitavastatin as an example, if you're using 1 micromolar, active uptake is going to dominate. But if you're at 10 or 100 micromolar, now the passive component is going to play more of a role. So this can influence, again, what may or may not be a rate-limiting step. So what are the influences of some of the phys-chem properties, looking at passive diffusion and the unbound fraction in drug hepatocytes? First, the graph on the left is looking at basically the log P, so looking at your passive diffusion component in relationship to your log D. So basically, your passive diffusion is increasing with the lipophilicity or log D, which I think is very intuitive. So we basically see the passive component increase proportionally with log D based on increasing permeability. Again, this is, I don't think, surprising to anybody. And now if we look at the fraction unbound in the cell, so now we're looking at the unbound fraction of drug that's going to be in the hepatocytes, and compare that in relationship to the log D value. So the fraction unbound of drug decreases proportionally with log D. This is due to a couple mechanisms. One is increased nonspecific binding. So as we increase the log D or log P, these compounds are going to have a propensity to interact with proteins and membranes nonspecifically. And also, as we increase log D or log P, we're also increasing the need to undergo metabolism in order to be eliminated. So between these two mechanisms, it's not hard to understand why the fraction unbound in the cell may be decreasing as log D increases. This can be taken a step further, looking at drug concentrations of hepatocyte and the relationship between passive diffusion and active uptake. Some of the more recent publications, particularly those from Brian Houston's lab in the UK, have used this KPU component, which is looking at basically it's the unbound hepatocyte to medium ratio. Can be more simply expressed, the KPU equaling the unbound concentration of drug in the cell over the unbound concentration of drug in the medium. This can be broken up into some basic components. If we assume that metabolism is negligible, so like in the presence of ABT, the amount of drug in the cell is going to be primarily contributed to two main mechanisms. One is active uptake as well as passive diffusion. And the drug in the medium in that relationship is going to, in the absence of efflux, is going to be primarily driven by your passive diffusion. So for drugs that have a high passive diffusion, your P-diff will dominate this equation. So compounds with a high passive diffusion, the concentration of free drug in the hepatocyte, it's going to be approximately equal to the concentration of free drug in the medium. It's not going to be exactly equal to, but it will approach that level. High active uptake, so for compounds that undergo active uptake transporters, such as some of the statins, like atorvastatin, the active uptake will dominate this equation. And for those, the free concentration of drug in the hepatocyte will potentially greatly exceed the concentration of free drug in the medium. Again, these are, I think, intuitively concepts easy to handle, but have been experimentally proven and modeled to demonstrate as much. If we look at drug concentrations of hepatocytes and how their reflection on metabolism, again, some of the more recent publications are looking at this cell to medium ratio. And if we look at the total drug, again, it's our concentration of drug in the cell over the concentration of drug in the medium. As I just mentioned on the slide before, we can do this also for the unbound drug, which is looking at the various processes involving getting the concentration of drug in the cell versus the concentration of the drug in the medium. One potential addition to this is if you have metabolism that is present and can't be considered negligible, you can factor in the metabolic clearance component into this equation as well. And you can determine the fraction unbound in hepatocytes by simply dividing the KPU over the KP total and deriving that fraction unbound in the cell. And in this slide, I'll show how these, with a number of compounds in one of these publications, how these different parameters move together and what the potential impact of metabolism is on the unbound fraction in the cell. So we have hepatic uptake and metabolism characteristics here of 8 drugs, many of which undergo active uptake. The first one I'll talk about here is at the top is atorvastatin. It has an active uptake clearance about 1,500, and the passive diffusion component is considerably less. Therefore, our unbound cell to medium ratio is quite high, almost a 500-fold difference. But if we look at our fraction unbound in the cell, it's fairly low. But one thing I'll point out is atorvastatin is also somewhat extensively metabolized. So this metabolism can actually deplete the free concentration of a drug in the cell. And kind of counter to that is kind of the opposite, where we have rosuvastatin, which again has a fairly high active uptake clearance and a fairly low passive diffusion. Here we still have a relatively high KP, so a unbound cell to medium ratio. But here our fraction unbound in the cell is almost 10 times higher than we saw for atorvastatin. Well, here metabolism is relatively minor. So in this case, metabolism isn't quickly depleting that free concentration. We're able to build up that free concentration potentially a little bit more, at least in relationship to the unbound fraction. For compounds that have a high passive permeability compared to their active uptake, like sequinavir, I showed this demonstration or these data before, that the active and passive components are somewhat similar. Therefore, our cell to medium ratio of unbound drug is now approaching 1. It's down around 2 in this case. In this case, the fraction unbound in the cell is quite low, particularly since sequinavir is going to be extensively metabolized. So we're really going, one is going to partition and not specifically bind, as well as a lot of the free concentration will be extensively metabolized. So the uptake as well as metabolism tend to go hand in hand. A recent publication that came out from a group at AstraZeneca last week, it was published in DMD, was looking at the effect of hepatocyte concentrations in incubation. This was in a particular regard to looking at metabolic stability experiments and improving the prediction of in vitro to in vivo for metabolic clearance of transported drugs. Basic result was your unbound cell to medium ratio decreases for actively transported drugs as you increase the concentration of hepatocytes in your incubation. What this kind of creates is now you have a lower concentration of unbound drug in your hepatocytes due to increased nonspecific binding and partitioning or creation of sink conditions when you have high concentrations of hepatocytes. This phenomenon was most notable for drugs that have a high unbound cell to medium ratio, so those that are particularly uptake transporter substrates, and the one that they illustrated here was atorvastatin. And the end result was that we had a decreased free concentration of drug in the cell. Therefore, we had a decreased amount available for metabolic enzymes, and what they observed was a decreased intrinsic metabolic clearance when the concentration of hepatocytes was increased in these incubations. Again, this was more pronounced for compounds that undergo active transport than compounds that do not, and they advocated for using potentially multiple concentrations of hepatocytes in these incubations, and particularly using lower, particularly 0.1 or 0.2 million cells per mil for incubation type conditions. So now I'll talk briefly about some of the scaling. So scaling our in vitro uptake and clearance data to scale it to the in vivo situation. For time's sake, I don't have time to cover definitely all the publications, but I'll highlight some that have been more recently described, at least in very general terms. Watanabe and colleagues had 2 publications in 2009 and 10 where they recommended using a scaling factor from rat in vitro hepatocyte uptake experiments to predict in vivo intrinsic clearance data. So what they did is they determined the scaling factor for rat for each compound that was conducted using human hepatocyte or rat hepatocyte incubations in vitro, and they scaled the rat based on the observed in vivo hepatic clearance. And from this, they were able to basically take these scaling factors and basically move it based on the uptake clearance and combine the metabolic clearance to predict the in vivo situation. Just to summarize this, the open symbols here represent a prediction of in vivo clearance from the in vitro data based solely on metabolism, so looking at metabolic clearance. And as we predict, particularly for compounds that undergo active uptake, the in vitro results underestimated or underpredicted the in vivo results. However, when they did this experiment with both rat and human using these scaling factors and looking at scaling uptake clearance, they're able to scale hepatic clearance from in vitro to in vivo much more accurately using these methods. There are other publications more recently that have looked at scaling factors and assessed inter-individual variability, particularly moving into human hepatocytes. So there is variation in these scaling factors that are needed to predict human in vivo hepatic clearance when we use these hepatocyte cultures in vivo, or in vitro, excuse me. So here they looked at various scaling factors that were needed across 3 different individuals of suspended hepatocytes. I believe it is, yeah, I can't. And with these 3 individuals, for 6 different compounds, they calculated scaling factors that would be needed to scale from in vitro to the observed in vivo clearance. Some simple things just to point out that even within a single donor of hepatocytes, depending on the drug and the differences in active and passive components, the scaling factor varied almost 20-fold within these 6 drugs. Now, if we also look within a same drug across 3 individuals, here for the case of repaglinide, we saw almost in this experiment, they saw almost a 10-fold variation in scaling factor that was necessary. Again, demonstrating some of the inter-individual variability. I think, again, that's somewhat intuitive based on our knowledge of the inter-individual variability and uptake transport from one lot of hepatocytes to the next. This was further confirmed, this inter-individual variability, in some other publications. One in 2011 observed a 30-fold range of estrogen 3 sulfate uptake into 14 different human hepatocyte donors. And again, another more recent publication looking at the variation and uptake clearance amongst 3 cultures of human hepatocytes from different individuals. The one thing that was most variable was the Vmax, and I think this is somewhat makes sense. We know, and this Vmax had then a direct impact on our predicted intrinsic clearance for active uptake, because our clearance is basically going to be our Vmax over Km. And this Vmax is going to be proportional to our transporter expression levels, so changes in this from our expression levels from individual to individual is likely a leading cause, potentially, of this inter-individual variability. This was also observed for repaglinide and rosuvastatin, as I demonstrate on this slide. And here I'm basically trying to show how this inter-individual variability, potentially in Vmax, can now change what you predict the contribution of active and passive components at different substrate concentrations. So here for both repaglinide and rosuvastatin, Vmax was variable, therefore our active uptake clearance was variable as well. What this results in are some of these graphs, as I demonstrated earlier, looking at the active versus passive components for these drugs. So this variation in Vmax leads to different predictions and modeling of the active and passive components, and how these components can shift depending on substrate concentration, depending on the Vmax and the uptake clearance that was determined with your in vitro experiments. So here, using one lot of hepatocytes with a low Vmax, another one with a high Vmax, could give you very different results and estimations of the relative contributions. There have been a number of studies that look at suspended hepatocytes versus plated hepatocytes. This one, a recent study here in 2012 looked at the uptake of numerous OATP-mediated substrates into 2 lots of suspension of hepatocytes done in suspension as well as plated. Here we see pravastatin is within 2-fold between suspension and plated within 1 lot. Within another lot, they are approximately equal. So there's some variability, maybe up to 2 or 3-fold, at least based on the results observed here. So if you're using the earlier time points for plated experiments, you're going to get typically, is what they're showing here, likely get similar results to what you see in suspension using the same lots. The same group also evaluated the protein expression of multiple OATP transporters in various formats, so looking at cryopreserved human hepatocyte suspensions, sandwich cultured human hepatocytes at day 5, and liver tissue from multiple donors. They looked at OATP expression. Generally what they observed is that in liver tissue is slightly higher than what was observed for OATPs in suspended or plated hepatocytes, and that in general there is little difference between suspended and plated hepatocytes when it comes to OATP protein expression levels. They also evaluated time-dependent changes in uptake clearance of various transporter substrates in cultured rat hepatocytes. So now just looking, as we extend these incubations out, not just from the first day, as we go out to 3 or 4 days in culture, here they observed for both torcolate, here in A, and rosuvastatin as an OATP substrate, that these decreased over time in cultured rat hepatocytes as we go out to 96 hours in culture, which is 4 days. The uptake clearance was also measured in plated human hepatocytes, so sandwich cultured human hepatocytes, again going from 5 hours to 96. And these 2 compounds, which were noticeably decreased in rat hepatocytes, there was some decrease in some cases in these. However, it was slight and definitely not as extensive as what was observed in rat hepatocytes. Another study from 2011 demonstrated the expression of OATP1B1 and 1B3 in human hepatocyte cultures, anywhere from 2 to 24 hours. And this was done by confocal microscopy. And as we start here on the left, looking at 2 hours in culture, to the middle at 6 hours, and to the right at 24 hours in culture, we're able to visually see the decrease in OATP1B1 expression in these hepatocytes. And that protein expression for 1B1 and 1B3 is highlighted below. Next, as we can see here, these results demonstrated about a 60 to 70% reduction in these transporter protein expression levels from 2 to 24 hours in culture. Now moving over to a little bit different, looking at some of the potential methods to phenotyping. I'll just briefly talk about the inter-system relative activity factors. So this was published by Hirano and colleagues in 2004. JPET, looking at the ability to determine the relative contribution of 1B1 and 1B3 to hepatic uptake, particularly using hepatocytes as a in vitro tool. Here they're able to evaluate 1B1 and 1B3 by using both cell lines that are specifically overexpressing a certain transporter, as well as looking at specific substrates for each of those transporters in hepatocytes and those cell lines, to be able to combine those relative activity factors together to then predict the relative contribution of 1B1 and 1B3 to the hepatic uptake of your test drug. So this was the relative activity factor, so they called this the selective substrate model. In the same publication, they talked about a second model, which is slightly different, looking at the relative expression model. Here where they're quantifying the protein levels in both cell lines overexpressing these transporters, as well as hepatocytes, taking the uptake rates of the test article, and basically modeling to predict the relative contribution of each transporter. There is quite a bit of data that is required from a practical standpoint to do these calculations. You'll need the uptake clearance of 1B1 and 1B3 specific probe substrates in hepatocytes and cell lines. For the substrate selective model, for relative expression model, you need to quantify protein or cell lines in hepatocytes. And for both of these, you'll need to evaluate the uptake clearance of test drugs into the cells. So while it can be done and useful, it does take a certain commitment to do these experiments. I'll briefly talk about estimation of the free intracellular drug concentrations. This is probably one of the more frequent questions that we're starting to get, particularly based on some of the scientific issues and the regulatory guidance that has come out. And using these free intracellular concentrations to predict drug clearance and potential for inhibition and induction of drug metabolizing enzymes and transporters. We know that it's the free concentration of drug within the cell that's available to inhibit these metabolizing enzymes or transporters, particularly efflux transporters, or cause induction via activation of nuclear receptors. Within the regulatory interest, the EMA in particular in the 2000 guidance discussed potential for estimating the unbound intracellular concentration when evaluating DDI potential with in vitro test system, particularly hepatocytes, particularly for those drugs that undergo active uptake into cells. And I think I've shown on some of the previous slides that the unbound concentration in the hepatocyte for compounds that undergo extensive active uptake can be significantly higher in many cases than what you observe in the medium. For time's sake and simplicity's sake, I'm not going to go into detail, but I've listed a number of publications here that estimate the intracellular free fraction, look at the unbound fraction in cells, which can then be used to essentially derive the intracellular concentration when combined with the free concentration of drug in the medium. It's definitely not limited to the following publications that I have here, but these would be good places to start. Many of them I have cited in previous slides. Some are more complex than others. Many take a very complex look, incorporating your uptake components, your nonspecific binding into account, your efflux or metabolism components. And there are many different potential ways to get there, again, based on some of the principles I discussed earlier in the presentation. So for simplicity's sake, I'm not going to discuss those in great detail today. There are some other publications that estimate the intracellular fraction unbound based on essentially surrogate endpoints, in particular looking at CYP inhibition or looking at transporter studies. And these are quite intriguing to use as surrogates that may alleviate the need to run some of these more complex uptake experiments and derive some of these modeling equations. Again, the utility of that is still somewhat hasn't been definitely yet refined. Now we just think some of the approaches to estimating cellular concentrations. What is the interplay between uptake and metabolism on the free concentration of drugs? And when we're looking at metabolism or uptake, what type of test systems would we recommend to use? If metabolism, our rate of metabolism is greater than our active or passive uptake into cells, at low concentrations of drug, it'll be metabolized relatively rapidly, and the free fraction will be low. And especially with high partitioning and binding compounds. Sequinavir was an example of this, where metabolism was significantly faster than the active or passive uptake. Therefore, a fraction unbound in the cell will be very low. In this case, hepatocytes also may be better predictors of clearance. If our active uptake is greater than metabolism, the drug can then accumulate, and this can increase the fraction unbound in the hepatocytes. Hepatocytes are going to be better predictors of clearance due to uptake transporters. Examples of these are going to be, again, your statins. And with passive uptake being greater than metabolism, your drug concentration will essentially equilibrate with media, as well as partition. Therefore, your free concentration in the media will be somewhat equal to what you'd see within the cell. So in these cases, subcellular fractions can do well at predicting clearance. With that, I'm just going to end with this slide. Just talked about some of the hepatocytes products that Xenotech currently has to offer that can be used for the conduct of many of these types of experiments. Two of these are suspension format, primarily our cryostacks and Hepatosure. Our cryostacks is a single freeze pool of hepatocytes, a pool of up to 2 or 20, and these can be used for the oil spin method or medium loss method, as well as our Hepatosure, which is our 100 donor pool. We also have attaching human hepatocytes as well as attaching non-human hepatocytes that can be used to do many of these uptake assays, as I've described as well. And with that, I will thank you all very much for listening in today, and definitely open up for any questions that you might have and try to answer a few as time allows.
Thanks, Dave. As a reminder, if you guys do have any questions regarding David's presentation, please remember to type out your question and send it in via the questions pane on the GoToWebinar user console. Before we move into Q&A, I would like to share with you all a few upcoming events where Xenotech will be participating in the very near future. For the sake of time, I am not going to read these to you. If you do notice that you will be in attendance at one of these events, please feel free to swing by the Xenotech booth and introduce yourself to our staff. So with that, I'm going to open it up to Q&A, and hopefully Dave has had a chance to review some of the questions, and we will be answering those now. Dave?
Yeah, there's a handful of questions, and I'll try to work through a few of those over the next couple minutes. One is, what is the typical concentration of ABT used to inhibit metabolism? It ranges. A typical is somewhere around 1 millimolar. It seems to be pretty common in many of these publications. We know that it does inhibit some P450s better than others. While it's a pan inhibitor, it doesn't completely inhibit all P450 enzymes, and it also has a limited effect on non-P450 enzymes in many cases. So depending on your routes of metabolism as well, 1 ABT may or may not be the most useful in some of these experiments. For the medium loss, another question, for the medium loss method during the centrifugations of also cause loss of drug from the cell into the medium. I guess theoretically it would be possible, particularly if you spun these hard enough and has some type of membrane integrity. And that is one of the practical challenges of these experiments, is actually the speed that you centrifuge down and be able to get these short time points and do them accurately. Another question, quantify the amount of drug in cells or medium. Is LCMS applicable in this case? For cold compound, yes, you can use, or even hot, you can use LCMS to evaluate the medium. When we're looking at clearance, well, what would be a reasonable concentration range for drugs in a study? Again, what we're going to try to do for these clearance types experiments is bracket KMV max. Many of these experiments will go anywhere maybe from 0.1 to 100 or up to as high as 300 micromolar. It seems to be common. It's going to be somewhat variable depending on your study design, your drugs, and those properties. For a single substrate concentration, for some of the basic methods, we tend to use a low concentration of substrate. either probably 1 micromolar or even less in many cases. Let's see here. Another question on the ABT. I think I answered this one a little bit before about ABT. Can we assume it has 100% inhibition of metabolic processes? The simple answer to that is no, and to what extent will depend on what enzymes are involved. And we have a few other questions, but for time commitments, I'm going to put those off. If there are specific questions that you would like us to answer, again, please feel free to email myself or anybody else here at Xenotech, and we can help you as you need. Thank you very much again.
Thanks, Dave. All right, before we close, I just want to share with you all a special product promotion that we are currently running. It is 40% off on all standard Xenotech products. This is valid on all orders of $3,000 or more. That's pre-discount. Use the promo code 40OFF on your purchase order or in the special instructions or notes area on the Xenotech eStore. This offer is valid in North America only and through March 31st. So this wraps up today's session. If you have any questions regarding any of Xenotech's products, please contact our Global Products Account Manager, Elizabeth Jackson, whose contact information is on your screen. Thanks to all of you for taking time out of your day to join us, and thank you to David for his wonderful presentation. Have a great rest of the day and week. Thank you for joining us.